Hi there, my name is Andrew Brown, and welcome to the electron transport chain. This video will lay out the structure of the organelle that houses the electron transport chain, the proteins with which the electrons interact as they travel down the chain, and the gradients and permeability factors that eventually result in ATP. After watching this video, you should be able to answer questions like this one, which tests your knowledge of the roles of the various proteins associated with the electron transport chain. Before we explore the biochemistry that occurs along the electron transport chain, let's first look at where it operates. Like some other components of aerobic respiration, the electron transport chain will occur in the mitochondrion of eukaryotes. To briefly review, the mitochondrion is composed of multiple membrane layers that increase surface area and allow for compartmentalized chemical processes. For instance, just inside the outer membrane, the inner membrane contains the protein assemblies needed for oxidative phosphorylation, the process whereby the electron transport chain produces usable energy. Folds in the inner membrane, called cristae, are what actually maximize the surface area and allow the membrane to support so many repeated units of the electron transport chain. Now that we can see that the inner mitochondrial membrane is packed with electron transport chains, let's take a moment to appreciate the interface of structure and function in the mitochondrion. As we'll explore in more detail later on, the electron transport chain uses a proton motive force to produce ATP. This force, which is essentially just a bunch of H+ ions, is stored in the intermembrane space. This will tie in nicely with other elements of aerobic respiration because, as the citric acid cycle is churning out NADH and FADH2 in the mitochondrial matrix, the energy produced from the transfer of their high-energy electrons can be coupled directly to the electron transport chain, driving the export and accumulation of protons in the adjacent intermembrane space. This incredibly synchronized process is what will, in the end, result in ATP production. Okay, so that was a lot of detail right off the bat. Don't worry though, that's the whole picture. The rest of this video will be devoted to expanding upon the processes already introduced. The way we're going to do that is by gradually looking more closely at the chain and then examining some high-yield elements. So, first, oxidative phosphorylation is accomplished by the sequential action of 4 membrane-bound complexes. They facilitate a series of oxidation and reduction reactions which transfer high-energy electrons all the way down the chain to oxygen. That point I just mentioned is worth repeating because it is an MCAT favorite. Oxygen is the final electron acceptor in the electron transport chain. Let's take another step back at this moment. Remember, the citric acid cycle is not the only source of high-energy electrons. Glycolysis also produces NADH. But because the mitochondrion is impermeable to NADH, a protein shuttle is needed to carry them across the outer membrane. We'll see a lot more on these shuttles in a little bit. When the chain is functioning properly, the high-energy electrons from NADH and FADH2 are used to create a proton gradient across the inner mitochondrial membrane. This gradient is vital to the electron transport chain's functioning in that the spontaneous exergonic process of the protons moving down their electrochemical gradient is coupled to the non-spontaneous endergonic process of ATP generation. So that was an overview of the entire chain. Let's now examine more closely the reductases and oxidases that compose the electron transport chain. The first complex houses the electron acceptor coenzyme Q, also known as ubiquinone. As NADH is oxidized, a compound called FMN, the flavin mononucleotide, will carry the liberated electron onto a cluster of atoms with slightly higher reduction potential, in this case, iron and sulfur. The reduced iron-sulfur clusters will then pass our high-energy electrons, originally from NADH, onto the final electron acceptor in complex 1, coenzyme Q or ubiquinone. This results in the expulsion of 4 protons into the intermembrane space, and the net reaction pictured here, which boils down this whole journey into the coupled oxidation of NADH to NAD+, and reduction of ubiquinone to ubiquinol. The second complex in the electron transport chain, succinate coenzyme Q oxidoreductase, utilizes the enzyme succinate, a dehydrogenase, to free up electrons. The molecule fumarate is also produced. Now, fumarate should sound familiar from the citric acid cycle, where it facilitates the production of NADH, the molecule that got this whole process started. This is just one example of how the various pathways in cellular respiration constantly feed forward and backward into one another. Okay, that was a nice aside, but let's return now to the pathway of electrons in complex 2. Succinate's electrons will be coupled to the reduction of FAD to FADH2 and then transferred to another cluster of iron and sulfur atoms. Just as we saw in complex 1, the iron-sulfur centers hold onto these electrons only transiently, before giving them to another molecule of coenzyme Q, which is the final electron acceptor for complex 2 as well. Notice that in each of these reactions, oxidation is being coupled with reduction. The two protons that are liberated from FADH2 are used up when ubiquinone is reduced to ubiquinol. Therefore, no protons are ejected from the matrix by complex 3, and conveniently, the net reaction shows only the oxidation of succinate and reduction of ubiquinone because the electron-bearing intermediates are regenerated along the way. The third complex in the chain, ubiquinol cytochrome C oxidoreductase, is characterized by the transfer of electrons from ubiquinone to cytochrome C. Cytochrome refers to a family of reduced ferrous proteins that, advantageously, can be easily oxidized. Because ubiquinone has 2 electrons to transfer and cytochrome C can only accept 1, 2 steps are needed to fully oxidize ubiquinone. In step 1, ubiquinol reaches its binding site in complex 3 by diffusing through the inner mitochondrial membrane. As just stated, 1 electron will grow to cytochrome C, while the other will be used to reduce another molecule of ubiquinone. Altogether, this first step results in the expulsion of 2 protons into the intermembrane space. Step 2 now will occur in a similar fashion to step 1. Following the arrival of ubiquinol and transfer of its first electron to cytochrome C, the second electron will then be given to the charged semiquinone intermediate from step 1, completing its reduction to ubiquinol. In addition to the 2 protons expelled as a result of the oxidation of ubiquinol, 2 more protons will be taken up from the mitochondrial matrix, which regenerates ubiquinol. So, after 2 cycles of complex 3, 2 molecules of ubiquinol will be used up, with 1 regenerated, to reduce 2 molecules of cytochrome C and expel a couple of protons into the intermembrane space. Okay, take a moment now to clear your head and refocus, because as I mentioned earlier, the fourth and final complex, which contains oxygen as the final electron acceptor, is likely to show up on your own MCAT. In complex 4, where we encounter the return of reduced cytochrome C from complex 3, individual cytochromes A sub 3, and oxidized copper ions are used to harness electrical energy for the eventual formation of water. As this happens, 2 more protons will be expelled from the mitochondrial matrix, resulting in the net formation of 4 oxidized cytochrome C molecules and 2 water molecules. Congratulations on making it through that. We're just going to review the specifics of the proton motive force and the NADH shuttles before we finish. The proton motive force, which has been alluded to before in this video, facilitates ATP generation from the electron transport chain. But how does it do this? As the intermembrane space becomes more concentrated with H+ ions, 2 things happen. The pH out there decreases, and the potential difference grows. Together, these 2 conditions create an electrochemical gradient, which, as the protons equilibrate, is used to fund endergonic ATP formation with ATP synthase. If ATP is one of the outputs from the electron transport chain, then NADH is an input. An NADH shuttle carries NADH not produced in the citric acid cycle into the mitochondrion, where its electrons can be stripped off and passed down the electron transport chain. There are actually 2 shuttles that can cross the mitochondrial intermembrane. The first is the glycerol 3-phosphate shuttle. We start with glycerol 3-phosphate dehydrogenase, a cytosolic enzyme that couples the oxidative regeneration of NAD+ with reduction of dihydroxyacetone phosphate to glycerol 3-phosphate. Before we go any further, dihydroxyacetone phosphate should have sounded familiar. It was one of those seemingly dead-end intermediates from glycolysis. Well, here it is showing up in NADH shuttling. In order for glycerol 3-phosphate to pass its high-energy electrons along, The shuttle makes use of a membrane-bound glycerol 3-phosphate dehydrogenase enzyme. Like its cytosolic sibling, the membrane-bound dehydrogenase couples redox reactions to convert glycerol 3-phosphate back into dihydroxyacetone phosphate and mitochondrial FAD into FADH2. Once reduced, FADH2 transfers its electrons to complex 2 via ubiquinone, thus generating the potential to eventually form 1.5 ATP per NADH via ATP synthase, all without NADH ever actually crossing the membrane. Another, more efficient NADH shuttle exists in the malate aspartate pathway, which generates 2.5 ATP per NADH. It starts with cytosolic oxaloacetate, a citric acid cycle intermediate, that also cannot pass through the mitochondrial inner membrane. However, with the help of malate dehydrogenase, an enzyme that couples oxaloacetate reduction to NADH oxidation, a product is formed that can cross the membrane. Once inside, malate is acted upon by a second malate dehydrogenase enzyme that catalyzes the reverse reaction, producing oxaloacetate and NADH. NADH can enter the electron transport chain directly at complex 1, but oxaloacetate must be converted into aspartate before it can be recycled. Aspartate transaminase catalyzes the interconversion of oxaloacetate and glutamate to alpha-ketoglutarate and aspartate, which can then cross the mitochondrial inner membrane and be converted back into oxaloacetate. To recap, in order to enter the electron transport chain, NADH from glycolysis must use shuttles. The actual ATP yield from cellular respiration is dependent upon the cell's efficiency. The pair of NADH molecules that take the glycerol 3-phosphate shuttle will make 3 ATP via ATP synthase, while those that take the malate aspartate shuttle will make 5. Therefore, combining the 5 to 7 ATP whose production will be funded by electron carriers from glycolysis with the 25 that will be produced from the carriers generated during the citric acid cycle, the total ATP generated from aerobic respiration will be between 30 and 32, depending upon which NADH shuttle is used. That concludes the material that this video will cover. Now let's put what you've learned into practice with some sample questions. This first one is testing your knowledge of the proton-evolving complexes in the electron transport chain. The answer should be D. This question is not as hard as it looks. Remember, there is only one complex that does not expel protons from the mitochondrial matrix. Either recall that it is complex 2, or recognize that D is the only answer choice here that lists 3 out of the 4 complexes. This next one is asking about the NADH shuttle mechanisms. The answer here should be C. A, B, and D are all narrowly false statements. In A and D, the complex numbers are switched, and in B, the ATP amounts are switched. If you got both of those questions right, congratulations. If not, please feel free to re-watch this video as many times as you need. Let's close with a brief review of what was covered in this video. We started with an in-depth examination of the electron transport chain and its membrane-bound complexes. Complex 1 liberated a pair of electrons from NADH in order to reduce ubiquinone and pump 4 protons into the intermembrane space. The net reaction shows a pair of protons also being added to ubiquinone as it is reduced. Complex 2 used an intermediate from the citric acid cycle as the electron source, but wound up with the same product. What is more, the protons liberated during this step are transferred to ubiquinone, rather than being pumped into the intermembrane space. Complex 3 needed 2 steps, because cytochrome C can only carry 1 electron at a time. And while each step resulted in the expulsion of 2 protons from the matrix, the fact that 2 more protons are used to regenerate ubiquinol explains why the net reaction shows only 2 protons as products. Complex 4, which expelled a mere 2 protons from the matrix, required 4 molecules of cytochrome oxidase to be oxidized to produce water. As far as the NADH shuttles go, if you remember one thing, make sure it's the ATP yields. The glycerol 3-phosphate shuttle will produce 1.5 ATP per NADH, and the malate aspartate shuttle will produce 2.5 ATP per NADH, so that, in total, 5 to 7 ATP will be formed as a result of glycolysis, and 30 to 32 will be generated overall. Thanks very much for watching, and good luck with your continued MCAT study.